The effect of drought conditions on Phaseolus vulgaris. Extreme variability in rainfall is very common in many Australian regions. As a result, these regions are put under high risk of drought, which can severely impact agriculture. With increased drought comes decreased yields, soil degradation, and crop failure. With agriculture producing $63 billion in 2019 to 2020, it is paramount that we understand how drought impacts the growth of plants. For this experiment, the common bean plant, Phaseolus vulgaris, was used. This was selected due to their fast growth rate, allowing responses to environmental stresses to be easily observed and recorded. Beans, among many other legumes, are commonly grown and exported by Australia. The aim of this experiment was to investigate the impact that drought can have on the common bean plant. This experiment was designed to represent Australian conditions, making the results relevant to the Australian agricultural industry.
For this experiment, we hypothesized that bean seedlings under increased stress through decreased water availability should have reduced shoot-to-root ratio and decreased chlorophyll fluorescence than the well-watered seedlings. This hypothesis allows us to observe how the seedlings may respond to increased stress that occurs in droughts.
The experiment was conducted using 8 bean plants. 4 were used as a control under well-watered conditions, as listed under the control, and 4 were under drought conditions and watered as displayed on the slide. Both plants were given nutrient solution on the first day. The plants were kept in the same greenhouse for the duration of the experiment to keep all other environmental factors, including humidity and carbon dioxide concentrations, constant between both plants. The plant's shoot height was measured each watering visit to monitor their growth. The measurements of the plant included comparison of the shoot and root, which was measured using the green weight by a scale, and height measured with a ruler. The root and shoot were separated from each plant before measurements took place. The instantaneous chlorophyll fluorescence was measured using a fluoropen analyzer by clipping the analyzer on the highest leaf and measuring fluorescence output.
On the left, we can see the visible difference of our samples. The control group samples have grown and appear to all be healthy, while underneath you can see that at the end of the experiment, the drought-treated samples have wilted and have brown, dry leaves. This demonstrates that there was an impact on the samples throughout the treatment. On the right is the data collected from the samples. Chlorophyll fluorescence, which is an indicator of photosynthetic energy in the leaves, was higher in the control group than the drought group. Shoot and root weight was also measured to indicate which parts were prioritized in the sample groups. As seen in the graph, the drought sample had a lesser weight of both parts compared to the control sample group. The ratio of shoot weight to root weight was also shifted.
A linear regression test was conducted for the shoot mass, root mass, and the chlorophyll fluorescence in control and drought bean plants, with no significant difference found in any of these variables. On average, the root mass was 0.7 grams heavier in the control plants. The shoot mass was 1.525 grams heavier in the control, and the chlorophyll fluorescence was 2,255 higher in the average in the control plants. The hypothesis that drought-induced bean plants would exhibit a decrease in root-to-shoot ratio and chlorophyll fluorescence was supported. Drought conditions negatively affected the chlorophyll levels in bean plants as there is less moisture in the soil, restricting vegetative growth and reducing the production of chlorophyll. The droughted plants averaged a lower chlorophyll fluorescence, indicating they were under more stress than the control plants. The average mass of both roots and shoots were also less in comparison to the control plants, signifying that the drought plants were limited in their oxygen, water, and nutrient uptake. The ratio of shoots versus roots was also lower in drought plants. This is because shoot length is dependent on the root's water intake to promote growth. All measured variables were not statistically significant, which may have been due to the small sample size and the overwatering of droughted plants on day 1 and 4. Further research into strains of bean plants that are more capable of withstanding drought conditions will be beneficial as droughts are becoming more frequent and intense due to climate change. The common bean plant has both nutritional and economic value. Additional knowledge would allow for more efficient agricultural practices leading to higher yields in Australia.